Yeah,
So it's here.
It's recording.
Oh,
Right.
okay, it's here.
There you are.
And how can I? It's here. And that. Okay. Is that? Is that?
Yep, we're good.
Okay. So today I'm gonna talk about the extracting rs-fMRI features for the neurological disorders classification and about the tutorial. And here is the table of the content. I'm gonna go through the data preprocessing and then brand isolation, connectivity, matrix estimation, classification, and feature extraction. So here's the website of the database I'm using. It's called LONI. It's Laboratory of Neuroimaging, and where you can find a lot of like neuroimaging data sets like acne, autism. What else? I don't remember. Okay, the data we are using is the structural MRI, which is core MPH, and also the functional MRI. And the format will be in DICOM. We have to convert it into NIFT. And here is for the rs-fMRI data preprocessing. And then all of the noise content will be in the script, which is Carlos provided before. And then it will be remove the first few time points and then motion corrections and then spatial smoothing. And then the fMRI to T1 weighted image registration. And we also apply the ICA aroma, global signal regression. And also finally, we will use the denoised fMRI data and then warp it to the MNI space.
Question.
I was going to say, I know why most of these steps are done because I've done resting state fMRI in the past. What does ICA aroma do specifically?
The ICA aroma is like they will detect and then remove the motion related noise.
Okay.
Yeah. Okay, but it's like decompose the fMRI data and also like structure the noise components.
Okay.
Yeah.
Okay.
And after that, we'll have the brain parcellation. And the brain parcellation, it will divide the brain into smaller regions, and the regions number is like depends on the atlas you are using. Like for example, this one is like AAL for this picture is like 120. And for each region, it will extract the average time series. Like so, it's kind of like the dimensionality reduction. Instead of calculating the voxel by voxel, it was just like calculating the RI to RI. But we have to check if all of the assets is in the same template space with the denoise data. And here is for the connectivity matrix estimation. The connectivity matrix is the representation of like different brain regions interact with each other. And then here's the example for connectivity matrix. We can use like precision matrix or also like correlation matrix to represent the connectivity matrix. And like for the rows and the columns, it's the brain regions, and then each cells is the strength of the connection between the different pair of the RI. Okay, so here is the most common method, which is Pearson, and it measures the linear relationship. It's better for linear data, but it also can be used on nonlinear data, but it will be better on linear data. Okay, it's fast, simple, and intuitive. The output will be the correlation matrix, which range between -1 to 1. The -1 means that like they have the strong negative relationship, and then the 1 means like strong positive instead, and 0, which means no correlation. But for the experiment, it measures the monotonic relationship. It can handle the non-linear data better compared to Pearson. And then the output would be the rank correlation matrix. It's also range between -1 to 1. And the Leavitt-Wolf shrinkage, it's a covariance shrinkage estimator, and it can regularize the covariance matrix to handle the higher dimensional data. It will shrink the sample covariance matrix just like normal baseline covariance matrix. It's just the normal covariance matrix, but they just call it sample covariance matrix. And they will shrink it towards the stable target matrix. And usually it will be the identity matrix. And it's also always invertible. And for the OAS, it's called Oracle Approximating Shrinkage. It's also the covariance shrinkage estimator. It's an improvement of the Leduc. They have more aggressive shrinkage optimized for the Gaussian data. And it outperforms Leduc. It can work on the small sample sizes data better than how Leduc perform. Because like for the regularization parameter used is don't laugh. it use like based on the number of the subject sizes. Okay, here's for the classification. The classifier I think everyone will be familiar with is the rs-fM, logistic regression, KNN, nerve-based, random forest. Oh, and here's for the feature extraction. For actually, most of the models can be used on this function, like the feature importance extraction. But here, for the example, I will just use the random forest. Because like for the each tree, they'll have to find the best features that can best split. Like which they have the lowest impurity score.
Yeah.
And then this is considered as the best split for each. Yeah, so for the
So conversely, for example, like with rs-fM, it would be the features that sort of help define
yeah,
the hyperplane.
for in classification.
Yeah.
The
Yeah.
support vectors.
So support vectors, yeah.
Okay, so
So each classifier has its own way to find those important features,
Mhm.
right? In this case, they need using the random forest because I think it's a little bit more informative in the way that you can know what the feature is doing, how it's doing the data. But yeah, like
It's probably much more intuitive too.
yeah, but yeah, support vectors, I mean, when you use SVM, there will be like some data points that are actually like the support vectors that are defining those hyperplanes that separate data.
Yeah.
Right? So, and yeah, if you use other classifier, each one has these metrics that you can use to identify which features are more important.
Mhm.
Okay, something that they needed to mention is like why she's doing this, right? And I think the important part of her project is that if you remember in one lab meeting, she presented a paper that was doing an evaluation on different ways to calculate the connectivity matrices and which one could be the best for fMRI data, right? The truth is that there are many factors that affect like the results. And actually in that paper, for example, Spearman was kind of the best metric to do classification. But when she ran her own evaluation, Spearman is not the best. Actually, like OAS and the UIT are way better than the other two, and also other metrics that she used. And the idea is that, well, you can use different methods and find these important features after classification, regardless of which classifier you are using. But once that you know which features are important, then you can retrain a classifier, and that can tell you if those features are actually relevant for the disease, right? Because in this case, the first run, she is classifying using all the regions, and then she get an accuracy. But after she selects the specific features, she will find out that the classification doesn't, I mean, the accuracy of the classification is really similar. So that means that she doesn't need to use all the areas, right? And that's kind of like a way to corroborate that those features actually are important for the disease. Right?
And it also simplifies future training.
Yes, I mean,
Reduces the computational cost dramatically.
yeah, it's a little bit of what, I mean, Spice started doing this with graphical lasso because one of the limitations that we usually have is that you have so many features in the data, right? We have 20,000 voxels in fMRI. So you cannot train a model using everything. So that's the reason why we first do the parcellation. But sometimes for new models, also even 120 regions might be like a big number. So you always want to find those areas that are more important. I mean, of course, you can find areas that are important for one kind of disease and different areas for other, and then you can do some kind of comparison. But I mean, she's going to show us how she doing this in code.
No, the what is
Well,
first?
First,
First, download the data. Here's the website. IDA. And here's the studies you can apply. And if you like some of the data set, if you really want to apply, you have to write the form and write the application form to them. And here's for the future. And you can select if you want a CT or MRI or PET or something.
Yeah, this website is really nice because they have a lot of different modalities of imaging for specific studies. And then you can kind of navigate and find out, for example, Bain is using ADNI and also AVIDE, right? From this website.
Yes.
One is for Alzheimer's and the other is for autism.
Yes. For example, here's the ADNI. ADNI, search and download. And here you can
Earlier, there are 2 subjects under 18 in there. Oh, the histogram showed 2 subjects under 18. Like, is that a data entry?
Well, maybe they have the for I mean they have kids.
It's for Alzheimer's.
Huh.
I mean, I just recently discovered that I accidentally had a couple of ages in my disease duration and had to fix some graphs. So it's entirely possible that that's a data entry error.
Yeah, all right. So yeah, this is the website. You can download data from here and yeah, you can explore that, right?
Yeah, here you can select like pre-processed data or post-processed.
Oh yeah, they also offer the original data.
But usually it's
Some of the
original.
processing.
Usually it's original. And here you can select which types of the projects you want. And then here is like research group. And then here you can show that the
Just
group
all
you're
of them.
finding. Yeah, here's whatever. And here is like MRI and fMRI. Usually I will select both. So we can download both that corresponding to each other. Yeah, here. Takes forever.
Right.
It's like,
I assume it's searching a large database.
yeah. Okay.
Oh, we got a lot of these guys.
Yes, you have to download the MPage, but I don't know why it doesn't show it here. Like most of the time you do that. Sometimes you will have some problem.
No, this is okay. I mean, eventually whoever is trying to download data
Yes.
needs to navigate in the website, right? So
Yes.
now you download the data and what you get out of it.
This is
So what is
selected.
what is the next step?
Oh, next step. You just download it.
Yeah.
And download it and denoise it. Denoise using the script you provided.
So you need to do the.
Oh, wait. Okay. Because it's all in DICOM format, we don't need that. We have to. Here. Here's the DICOM. It's an app where you can convert the DICOM to nifc. Yeah, here is where you put your output file name. And here is the directory for output. And here's the format we want. Where the nifc, you can work with FSL. And then you can do the visualization. And here is like for the metadata. And for this one, I will set it as default. And it's like about the intensity out of the thing. And here, just select the folder and then you can convert it to Nifty.
All right, that converts data from DICOM, so it's the format that the scanner gives you to the NIFTIs, that is a compressed format for analysis.
Yes.
Okay.
Here. Okay, here's for the example of the atlas we are using, and here's the denoise data. And here's the atlas. You have to check if it's in the same place with the denoise data. Here's the, yeah. Here's different atlas I've tried to use before. So close it. Close it. Yeah, that's all.
Okay.
Okay, I have to close it. Here's my code. So many things. Okay, here's for the code. Here's where we import the libraries. Yeah.
So what is the main library that you're using?
The NumPy.
NumPy, pandas, is the regular stuff, Seaborn,
Yeah.
and then you are using Skylearn for the models.
Yeah, this
right?
one is like for the precision metrics. And here is for the classifier. Yeah, this is also for a classifier.
All right.
And for the brand facilitation part, we have to define the RI numbers first. And here's the visualization of our atlas. I use the AAL for example. And then we will use the Nifty labels masker. And then here is where you put the atlas. And then we have to set the standardized as true. And then here's the masker. Just generate the masker. And here's the picture of our AAL. And then, oh, I just use one example for here. And then here I use the get subject data. And I will mask this data using that masker. And then normalize and standardize it. And then here's after using, how do I say it? After using the brand isolation, and then here's the how the array looks like. And now I
So that array is the mean time series of
Yeah.
each regions on the part on the atlas, right?
yeah,
Okay.
like if
In this
120,
case, 120.
that means that they have 120 of the mean
Mhm.
values of each
And
regions. And
176
this is the time
time
point.
points.
Yeah.
Okay.
And after that, we will have to compute the connectivity matrices. And here is where I import the data set. Here I use the SCAR7 data set. Yeah, here is where I load the data at these two. And here is the shape of the data set, like the size. Here is for the RI number, as we said just now. And here is for it.
So everything is concatenated.
Yeah, everything is concatenated. We have to split it afterwards. And then now we have to check how many subjects we have. So it's like divided by the time points value. Like for example, a single subject data size is here. Where is it?
176.
Oh yeah, it's 176. And for this one, if example is 170, and then I have to divide it by 170. And then we can get the size is 57 for patients and 34 for the controls. And for getting the precision matrix and for the ladle because the ladle and OAS is just now as we see, like you extract the covariance matrix. And if you want to find the precision matrix, it's just like the inversion of the covariance matrix. So you just fit it into the model and then extract it. They have the built-in function. And for the Pearson and Spearman, we just have to extract the correlation matrix. That's all. Yeah, this one is same as just now, but all of the data is concatenated. We have to split it like using this one. And then we have the data size number just now. just split it and then reshape it. Oh, not reshape it. It's just like they will find the precision matrix one by one for each individual, and then we'll concatenate it at the end. Okay, so here is like I just use the Pearson as example, and for 57%, we will have their own precision matrix, same as for the controls. And here is for the part for classification, because all of the precision matrix is symmetrical, so we only need the upper or either the bottom, and we don't need the diagonal. So here is for the one.
The upper triangular part of the matrix.
Yep. So here is for the one for extracting the upper part. First, we have to reshape it to the, because it's like symmetrical, as I said just now. So we reshape it back, and then for each individual, we have to extract the upper triangular part. And k equals to 1 means that it exclude the diagonal. Here is for the classification. We use the random forest. And here's for the test data, we random sample, and then the trend data is like the data excluding the test sample. And then we'll extract the upper triangular part, same as this one. And for this one, we'll feed it into the random forest classifier. And then this one is like the built-in function as well. If you want to extract the feature importance, you can just call the model and then extract the function, extract the importance. And then I will calculate the accuracy as well. And then I rank the features based on the scores at the end. And then after that, I run it. And then here is for the features. And here's the accuracy. And then because we want to know like which region, because it's like the connectivity, we want to know the real ROI in each indices. So we have to map it back. For example, I use this one. And then here is the atlas indices, like the labels we are using based on the AAL. So here is, we have like 120 labels, and then we have to map it back. So for the indices, we have to map it back like to 120 and 120. So for the each row and each column, we like just divide by 120. And then here is the tuples, including all of the pairs of the RI. And then here we will map it back using this all column labels. And then here is the result. That's all.
It's basically the connections that are more important for distinguish between patients and control.
Yes.
All right.
So feature number 6,000, you scroll up slightly. 6,905 corresponds to,
Yeah, 57.
is that subject number 57, or no, those are two different ROIs. So ROI 57 and ROI 65. Okay, and then that is the
Yeah, and this is-
occipital,
yeah.
the right inferior occipital and right inferior parietal.
Mm-hmm.
Nice. All right.
Let's stop it.
Questions on online?
No questions.
I'm good. Thank you.
Yeah, I'm good.
All right, super clear, I guess, for everybody in the room. Jason, any other questions?
No, actually, this is possibly the one that I'm most comfortable with because it uses a lot of the tools that I've used before.